Hello, everybody. Unfortunately, I can't be there with you today because my kids are sick and can't go to daycare. But in an effort to not lose track of where we are and not losing that much time this week, I'm going to do the lecture with you through this video. That way you can follow along, pause whenever you want, take some notes, and after this quick lecture, we'll review the toothpick ace lab that we did on Friday, which will set us up nicely to do another lab tomorrow. So please, as you get into class today and pull up the Google Classroom assignment, when you click on this, get your notes out, start taking notes on the video, and then you can finish working on the toothpick ace lab as the video concludes. And if you have homework to do after that and that you have not finished yet, you can continue working on that as well. So we'll get started here with the enzyme lecture then. With enzymes, what we were originally talking about with proteins is really going to apply here really easily. Um, we talked about on Friday how proteins were a large, big umbrella category of molecules, and then that under that was a group called enzymes. And the enzymes are going to be machines, molecular machines that do jobs in your body and change one thing into another, basically. What is going to be the structure of an enzyme? The structure of an enzyme is going to be the exact same thing as the structure of a protein. Enzymes are going to be made of amino acids, and those amino acids are going to be put into a very specific order. That order is going to dictate how the enzyme folds up, and once it does fold up, that'll give it a definite structure, which will then give it a job. In order for an enzyme to work, all of the amino acids that are part of that enzyme have to be put together in the exactly correct way. If they are not, the enzyme will not work. Okay? Um, and so we get to this drawing here, this diagram. This diagram does a pretty good job of showing you how an enzyme might react with a molecule. Okay, when we take a look at an enzymatic reaction, it's really important to take note of a few things that are going on here. An enzyme is always going to be a protein. And where that enzyme reacts with its molecule is always going to be called the active site. Okay, that's where the enzyme is going to bind with the molecules it's interacting with. Those molecules are going to be called substrates, and they can come in many different forms. Okay, when all of those things come together, the substrate bonds with the enzyme at the active site, you'd call that a complex. That complex is where the reaction is going to take place. Once the reaction does take place, energy is either going to be absorbed or released. And when that occurs, a product will be released, and the product can then go off and do whatever it's going to do, leaving the enzyme empty. This enzyme, and this is a really important piece to this enzyme can now be reused and go right back to the beginning here, where it can touch some more substrates, interact with them, and form more products. Enzymes are reusable. Please write that down, okay? Um, and so in looking at this reaction, what you're seeing here is a condensation reaction, okay? Take a look at how that applies to this. This enzyme is going to react with these two substrates. These two substrates are two small amino acids. The complex, as it forms, is going to absorb energy and form a bond between those two substrates, releasing water. That was the definition of a condensation reaction. Releasing this forms a dipeptide, and now this molecule is going to be the beginning part of a protein. So this enzyme, which is a protein, is making another protein. Okay. The notes here at the bottom pretty much say the same thing. Enzymes bind with substrates at active sites. Once they release their product, they can be reused to perform the same reaction. Okay. I did also put here at the bottom that this is an example of a dehydration reaction. Oops, sorry there. Can't see. Um, but you could also call it a condensation reaction. That is the same thing. Okay. All right. Moving on here. How do enzymes help speed up reactions? This is going to be kind of essential to why your body uses enzymes. Okay, so let's think about chemical reactions all together now. Chemical reactions involve reactants that turn into products. This can either be in the form of a condensation reaction where two small things are being connected together to make one big thing. Or it can be the reverse, where one big thing is being broken down into two small things. Okay, whatever it is, a chemical reaction is going to involve taking a reactant and turning it into products. And essentially, energy is going to be needed to get that done. If you were to look back at this example here, pretend that the purple enzyme is not there. Try and just ignore that. Just focus on these yellow substrates. How much energy would be needed to actually take these two substrates and connect them together without the enzyme? Essentially, that could happen. However, these substrates would have to be moving so fast that they slammed into each other like a car crash, or like an asteroid hitting another planet or a moon or something. These two substrates are going to be having to move so fast and colliding so precisely that a bond forms in between the two amino acids. Okay, that is the amount of energy needed to turn those reactants into products. So you can theoretically look at this and say, well, could these two substrates turn into this product without the enzyme? Yes, but they would require so much energy because they'd have to be moving so fast and hit each other precisely in the right spot that, the amount of energy required here is just too much to supply to get this reaction done. Your body would have to produce so much energy to get this one simple reaction done that it really wouldn't be worth it for your body to do this. So, how do enzymes help? Enzymes help by lowering the amount of activation energy, which is the amount needed to get started. They lower the amount of energy needed to make this reaction happen. So what is an enzyme going to do? Well, by having this enzyme in your body bind with these two substrates and then connect them together, it lowers the amount of energy needed to get this product made. Now these substrates don't have to be moving at an incredibly fast speed to produce this product. They just have to bump into this enzyme, and the enzyme will supply the rest of the energy needed to, well, won't supply energy, but this enzyme will do the work of combining these two substrates into a product so that the body doesn't have to supply such an incredible amount of energy to get it working. Okay, so that's essentially how chemical reactions are going to occur, and enzymes, really important, this last point here is important, enzymes will lower the amount of energy needed to make this reaction happen, or make any reaction happen. Okay, this is what I just said in graphical form. Okay, so here's a reactant, or two reactants, and a product. Okay, in this reaction is going to release energy, and so in essence, this reaction is going to release some kind of heat. All right. If the reactants have more energy than the products do, the amount of energy released is usually released as heat. To turn these reactants into these products, though, energy is needed to get started. These two things have to slam into each other to make this. How much energy is needed to get these two things to slam into each other and make that? A lot of energy. way more than with an enzyme. So without an enzyme, you've got to slam those two things together and, oops, sorry, and hope that the product gets made. But with an enzyme that's precisely engineered to combine these two things right here, this enzyme can do that with a little energy required so that the body doesn't have to supply so much energy to make these products. Okay, so this is, again, the same thing as these notes, just in graphical form. Okay, the last thing we're going to look at here are some examples of enzymes and then what kinds of things can affect them. We've kind of talked about one of those already. Here are some examples of enzymes. The first one's protease, that's going to break down proteins. Kinase is going to deal with phosphate molecules in your body. Salivary amylase is going to deal with breaking down carbs in your body. All of these end in ASE, and you're going to notice that that is a trend with enzymes. Any name, or if you see anything named with ASE at the end of it, that basically means it's an enzyme and it's going to perform some chemical reaction. So what kinds of things can affect enzymes? Well, we talked about this last week and how enzymes have a specific shape to do a specific job. If you change the shape of the enzyme, you change its job, and basically its new job is it's broken. It has no job. So you can either change the shape of an enzyme by changing the amino acid sequence, which will definitely mess up how it folds into a 3D protein, or you can put it in an unusual environment. And by doing that, the protein or enzyme will fall apart. Some things that can denature an enzyme: temperature, heating it up, changing the pH of the environment it's in, or making the enzyme more acidic, right? Changing the amount of salt or salinity of the solution the enzyme's in can change its shape. By changing the enzyme's shape, you lessen its ability to do its job, okay? This is a pictorial way of looking at this too. If the normal protein has a very specific looking shape and then you heat it up, its new shape will be so unrecognizable that it won't be able to dock with the protein it's supposed to, or dock with the molecule it's supposed to interact with. Okay. Last thing we'll look at here is feedback, and this really pertains to our toothpick lab pretty well. Okay, so there are two things we're going to look at here. One is feedback, and one is inhibition. So we'll just focus on the left-hand side of this slide first. The first thing we'll look at is inhibition. Enzymes can be affected by the molecules around them. Some molecules can be inhibitors, which means they get in the way of the enzyme and its work. This slows them down. Let's take a look at what that could be, right? In inhibition, enzymes are going to bind with a substrate. That's the normal reaction. But if a competitive inhibitor is also in the environment, it might sit right in the active site and block the substrate from getting in. That's going to slow the reaction down. In a non-competitive way, a non-competitive inhibitor or some other molecule might dock to a different part of the enzyme and change its shape. If the shape is slightly different, it might not be able to interact with the substrate. This again would slow it down. A very easy example of this would be in your toothpick lab. Competitive inhibition is going to be really easily modeled when we think about the round and flat toothpicks trial. There were in that trial round and flat toothpicks, and you had to feel the shape of the toothpick before you broke it. If you picked up a round one, you had to drop it and pick up a flat one in order to break it. That got in the way and, I presume, based on your data, slowed you down. Okay, so this is a type of reaction that can occur with enzymes. The other thing we're going to look at, and this is on the right-hand side of the page, is called feedback. Feedback is a concept by which enzymes can be affected by their own products. If an enzyme's shut down by its own product, that's called negative feedback. If the cell makes more of a product based on the reaction that's occurring, that's called positive feedback. Let's take a quick look at, um, well, and you know what, this one, this diagram is a little hard to describe if I'm not there. So we can just, let's focus on a different one. Okay, let's focus on something else. Can I write this? No, I can't. No. Let's see if I can look up one. Since all right. Well, we'll use this as a quick example of what I am trying to get across to you. Doesn't really look like a very good picture here. Can load a different. Well, we'll try and use this picture right here so we don't waste too much of your time. Um, all right, so your body maintains a certain amount of blood sugar, and this is one example of homeostasis. If you were to eat something like a cupcake, the amount of sugar in your blood would go up. Your pancreas would sense that and start making a protein called insulin that goes into your blood, pulls that sugar out, and then returns it to the cell, lowering the amount of sugar in your blood. As soon as that gets done, that protein stops working. That's called negative feedback. And so, again, what's happening here is as the amount of sugar comes out of your blood, as that starts to happen, that shuts off the making of more insulin, which stops the reaction from occurring. And so that's an example again of negative feedback. Here would be another kind of example of this. Let's say you didn't eat breakfast this morning, and so the amount of sugar in your blood's really low. Your pancreas is going to sense that, release a different enzyme. This is called glucagon. That's going to go into your liver and release some of the stored up sugar you ate earlier. That's going to raise the amount of sugar in your blood, and as that sugar goes into your blood, that shuts down the making of glucagon, which would continue this process. And so that's a very quick example. I know it's not, you know, what this was, but I think that's actually going to serve us a little better here. So in this, to finish this off here, feedback is just that. The products of the reaction shut down the making of more of that product. And we didn't actually go over an example of positive feedback, but we can talk more about that maybe when I get back. Negative feedback here is going to be the important one to focus on, especially for our lab. Okay? So then, right before we leave here, and I know we're almost out of time, let's take a quick look back at your lab, okay? I wanted to review this with you in class, so since I can't be there, let's just do it now on this video. The first three questions of the lab ask you to think about the different parts of the reaction. So if toothpickase, U, was the enzyme, the substrate here is going to be what you're reacting with. And if you put down flat toothpicks, you got that right. Okay, flat toothpicks was the substrate or the first thing you reacted with. The active site is going to be your fingers. Okay, that is where the reaction is occurring. The product should be broken flat toothpicks. If you then think about these questions below, and they shouldn't be too hard to think about here. You're thinking about which reactions have the highest and lowest enzymatic reaction rate. So it just means in which trials were you fast at breaking down the toothpicks and which trials were you slow. The highest reaction rate is probably going to be the trial with no inhibition or no denaturing. So trial 1, where you could just break the toothpicks, right? The lowest reaction rate might be either trial 2 or 3, because in trial 2, round toothpicks were getting in your way. That's competitive inhibition. Or your hands are being taped in trial 3, which is an example of denaturation. Okay, um, that kind of explains question 6 here. And then I think you guys can answer questions 6 and 7 actually now based on the talk we just had. Um, the last thing we should look at here, though, is just thinking about, how enzymes affect the making of products. Okay, these last two questions really get you to think about what would happen in different scenarios. So let's just focus on this first one. Explain what the graph would look like if there was no enzyme acting on the toothpicks. You might guess then that the reaction would look really silly, or the graph would look really silly, right? You'd have a pile of toothpicks in front of you, and you'd just be staring at them. No one would be reacting with them. So what would happen? No reaction, okay? You'd have to supply a lot of energy to get those toothpicks to be broken without an enzyme. And that could be heat or motion or something. But, a lot of energy would have to go into that to get any of those toothpicks to break. So presumably, there'd be no broken toothpicks for question 8 here. For question 9, though, think about, okay, there's 2 students working on breaking down this pile of toothpicks. What would that look like in comparison to 1 student breaking down the pile of toothpicks? Well, if there's 2 students doing it, or 2 enzymes, it would happen faster. And, this gets us to thinking about enzymes and how they react with the substrates, right? If you think about an enzyme as a worker, or as again, a machine turning one thing into another, then if you get more enzymes working on the exact same problem, or the exact same, making the same product, then the more enzymes working to make that product, the faster it will go. On the contrary, you might think, okay, well, if there's one enzyme and I want to make a lot of product, let's say I want to make a lot of broken toothpicks, if I give that enzyme 10,000 toothpicks, is the enzyme going to work any faster than it did when I gave it 50 toothpicks to work with? The answer to that question is no. Enzymes only work as fast as they can, even if you give them more product to work with. Okay. The last thing I want to mention here is the law of conservation of mass. We can talk a little bit more about this tomorrow, but this wasn't a direct question in your lab, and I did want to address it in here. Um, enzymes are agents in speeding up chemical reactions. They turn substrates into products. It's really important that you realize that even though substrates changing into products changes what those molecules look like, that none of the mass is lost from beginning to end. Notice in this example, this enzyme takes in 2 substrates, combines them together, and makes a product of the same mass. Um, and so in doing that, no mass is lost. Energy is utilized to make this reaction happen, but no mass is lost. The same amount of mass that was at the beginning of the reaction is also at the end of the reaction. Okay, that is called the law of conservation of mass. We'll try and address that tomorrow as well. Okay, I hope this video helped out in explaining a few things. Tomorrow we will do a lab that demonstrates all of this, and then on Wednesday we can review some more before we take a quiz on enzymes, okay? Hope you have a great day. Thank you for listening, and I'll see you tomorrow.